Liver disease is a huge problem worldwide and is responsible for a staggering 2 million deaths per year. If you average this out, it means that in the time that it takes me to give this short presentation, 11 people will have died of complications associated with the disease. Now, although there are a number of different types of liver disease, what they all have in common is if left untreated, they result in fibrosis, which is an excessive scarring of the liver. And eventually this leads to cirrhosis, where the level of scar is such that the liver can no longer carry out its vital functions. Now, most of our understanding about the mechanisms that underpin fibrosis have come from work in rodent models, and there remains this huge translational gap between potential therapeutic targets and effective patient therapies. To put it into context, currently there are no approved antifibrotic therapies to treat patients, and transplant is the only option. So how do we solve this problem? Well, we need to understand the human disease. We need to understand the mechanisms and the cellular players that regulate human liver fibrosis. To address this, our lab uses a technique called single-cell RNA sequencing, which allows us to gain valuable insight into the transcriptome of individual cells from healthy liver and cirrhotic liver, and in doing so, aid in the identification of important cell types. Now, the way that I like to describe single cell and its strength is to imagine that you're interested in understanding how a research lab works. Now, you could characterize everyone in the lab as one and identify them all as scientists, which is true, but you're missing out on important information. If you look at each person individually, like we do for a cell in single-cell RNA sequencing, you find out that we have postdocs, we have students, we have PIs, we have bioinformaticians. Each one of these groups or populations are scientists, but they also have quite unique and specific roles in the lab. And this is no different to cells in the liver. Because an ideal antifibrotic would target only the cells that are responsible for fibrosis and not the homeostatic ones, we focused in on the identification of these cells in the cirrhotic liver and identified a population of scar-associated macrophages, scar-associated endothelial cells, and collagen-producing myofibroblasts. In the cirrhotic liver, we find that the location of these cells is within the fibrotic niche, in close proximity to one another, as you can see in the image depicted in panel B. Now, these are the cells that we want to be targeting. If we focus in and home in on these populations, we can actually start to interrogate the ligand and receptor interactions between them, as shown in panel C. And in doing so, we're taking the first steps at identifying new targeted therapies to treat chronic liver disease. Thank you.